Hello everyone, I'm Yuan and welcome to my channel of Lighthouse Pharmacology. In this video, I'll be talking a little bit more about bioequivalence study, right? So it's like the third video on the bioequivalence topic. So whereby in this video, I'll focus more about the washout period. How is it being designed? How long is the period sufficient to be considered a sufficiently long enough washout period so that there's no carryover effects from the previous treatment. The second part will be on the characteristics to be investigated. The main characteristics in a way in this context actually refers to the data or the pharmacokinetic parameters that you need to collect to enable you to do your final calculations, whether it's BE or not. And the third part will be actually on the strength to be investigated because most of the manufacturers will actually produce more than one dose for that particular drug. So is it necessary or required by the guidelines to do different BE studies for different doses, or under which circumstance that you actually need to do different doses for the same drug, or one is actually good enough that the data or the BE status can be extrapolated to all the other dosage variants. So the main reference for all these three sections for this particular video is actually based on the EMA guidelines 2010 and also the ASEAN 2015 guidelines. So the EMA obviously is the European guidelines. So if you're actually from not from the ASEAN country region, but you're based in another country which also heavily depends or relies on the EMA guidelines as the main reference, I hope that this video could be useful for you too. All right. In this slide, we will be looking at a little bit about the categorization of the different drugs available in the market based on their formulation. So there are three main categories. As first one is the modified release, the second one is the immediate release, and the third one is on the other etc., such as effervescence, orodispersible, and so on and so forth. So there are clearer guidelines that actually dictate the details of it. Right. Although this is actually a table or a summary by the Malaysian guideline for the drug registration guideline 2022, but most of the guidelines like the EMA or ASEAN also follow the same categorization as well. So for the drugs that we're going to discuss about for this particular video, most of it actually falls into the immediate release form, but there are others like as mentioned, like the modified release and orodispersible one. Some of the details will also appear towards the end of the video as well. Let's start with the first section of this video. Remember, there's three sections, so we're going to talk about the washout period, the second one, the characteristics, and the third one, which is on the strength. So about the washout period, as we all know, it's like the analogy like a washing machine in a way. Our body will actually eliminate or excrete the drugs, right? So and most of the BE study, when you look at the design of it, this is just an example for a typical crossover study, which most of the drugs will actually use this particular design just because that you can use back the same participants, but you just give them the particular tablet A, the original, or B, the generic tablet for during a different week, right? So before the person actually comes in for the second time into the BE center to get the second dose, either it's a generic drug or the original one. So we as a study designer, we have to make sure that whatever drug that has been taken the first round has been eliminated almost completely, right? Obviously we don't really measure whether is it 0.00, but it has to be low enough and it's out of the system, most of it, so that it doesn't bring an carry over effect, so that the second dose that you take is not a 1+1, so it has a 1+0 baseline, so that your data would be comparable, right? So bear in mind, the main objective of this whole study in a way when you're designing it is to be able to generate this particular graph in the end, right? So whereby you can compare the AUC between the different graphs, whether it's the Biowaiver concentration or the generic drug concentration, and you can get hopefully comparable or similar Cmax or Tmax and so on and so forth, then you can happily make a conclusive evidence to show that, hey, this is BE. Right, so to able to get this, so as you can see, so both drugs, you need to make sure that you actually start from 0. So to enable this to occur, you must make sure your washout period is a good timing, right? Next, when we look at the standard design, right, so a lot of it of the standard designs are a randomized, so it's as a 2-period, 2-sequence, single dose crossover design. So again, the washout period, we need to be sufficient so that the lower limit of bioanalytical quantification will be, as I say, like the graph just now shown, it'll be starting at 0. So we normally try to make it, if you want to by calculation, it will be at least 5 half-life, elimination half-life to do it. So again, the purpose is to, as mentioned in the text and mentioned just now, is to prevent the carryover effects that can interfere with your results that make your data invalid. In this case, we'll actually be looking a little bit more on the alternative design. Remember, the one just now is actually under the standard design. So now we look at the alternative. As we all know that the different drugs, they all behave differently. So under very rare circumstance that we might need to use a multiple dose and things like that, so that in certain cases there'll be like steady state concentration because it's a multiple dose. So the washout period, again, we'll try to get at least 5 terminal half-lives. Yep, so but anyway, if you want to use a different design compared to the standard ones, you must make sure it obviously has to be properly justified, right? Why you have to use such a complicated design. So there's only very rare circumstance that it probably is due to limited sensitivity of the analytical method, right? Which is very, very rare again, because there's a lot of advancement, you know, on the development of sensitivity of HPLC, UPLC, and things like that you can use, right? So that you most likely avoid this, right? So again, it can be accepted as well if you have to do multiple dose, again, because we are administering it to human subjects or human volunteers. Obviously, we need to make sure that it doesn't cause very nasty side effects or it's tolerable for them. So when do we use it? So hopefully it's already given you some of the information. So under very rare circumstance. So when do we know which drug to use? So a simple way, as will be mentioned again in the conclusion, is that you just have to check the literature, right, to see what people has done on that particular drug, to give you a better gauge about it. Because if you want to show the authority that you have to use all this more complicated alternative design for a specific drug, you actually have to provide evidence as well to show that, the single dose way just couldn't give you the results. And it's impossible to get bioequivalence data, right, due to scientific reasons. Let's look at another scenario whereby the washout period is a little bit more tricky to calculate, i.e. it's not as direct as the 5 half-life, which is about endogenous substance. So this is another field of its own whereby there's a lot more tailoring, when obviously the more popular ones will be the hormonal related ones, as hormonal replacement therapy and etc. stuff. So it cannot be directly evaluated because obviously everyone has an endogenous baseline level of that particular hormone, so you can't say that the baseline level is a 0. So there's extra care to make sure the washout period is sufficient, right? And bear in mind, most of the time that we do endogenous study, because we are doing it in healthy volunteers, for example, so our level could be higher, obviously, will be at the normal higher level, right, compared to those who are actually require treatment for that particular drug. So there's tailoring of doses and so on that we can see more details of it in the later slides when we talk about the characteristics to be investigated. Well, in this particular section, we'll go through the second part of the video, which is on the characteristics to be investigated. So whereby in this particular context of BE study design, characteristic doesn't mean a lot of fancy stuff, how the drug behaves in a way, of course. So how the drug behaves is reflected into their different pharmacokinetic parameters. So again, for BE, there are three main parameters which is used as a guideline in a way to determine whether is the drug BE enough or not. So which is the AUC, Cmax, and Tmax. So, but again, bear in mind the different drugs are different. So how do we need to actually tailor make some of the study design to be a little bit more geared towards the drug that you're trying to measure? It's all stated very clearly in the guidelines as well, which we're going to look through in the next few slides. So what info do we need to collect? In a way, as mentioned, the three main parameters. So, and if it's the AUC is to infinity, so we try to cut off at 72 hours as mentioned in all the guidelines, and it's also mentioned in the earlier video as well, right, whereby the 5 to 6 half-lives in a way, most of the time it should be end at around 72 hours. So the the cutoff point is at 72, right? So the guidelines in a way, it's not to make life difficult in a way, difficult for the manufacturers, right? But it's also try to make it simple for them as well, but also we should not compromise on the quality of data, so we try to measure it in the most accurate way as well. So we try to get a balance between simplicity and also accuracy. So before we move on to the separate later sections which talks about the different categories of drugs in terms of how we can actually design our study to fit into or to obtain the data of the characteristics that we want, there's also some general guidelines at the top of the section on this particular section, which actually mention that the feasibility of the usage of urinary data if you really need to use it when you're running out of options in a way. And another part is to assume that we'll be using the concept of non-compartmental models in our calculations. So as a short or brief reminder, non-compartmental models, the main concept is we are assuming that a person, it's actually like a conical flask, right? So if you take a tablet, it's like, i.e. you drop a tablet into the flask, which has fluid, i.e. it's like blood, and whatever body fluid that we've got, and it will disperse evenly across different tissues, right? So obviously the concentration is something that we get from the blood directly, right, from the BE study, the sampling, right, but we actually exclude all the possibility of first order, second order kinetics and so on, which is going to make the whole calculation more complicated, right? So we're just going to use the overall simplistic model of non-compartmental in our calculations. I think you've seen this slide before in the first video on bioequivalence. So this is just a reminder that every drug is different. So we try to categorize them like the ones that you've seen earlier on, like some of it will be like endogenous substance, right? Later on we have more categorization of the drugs that we're going to go through under this particular section. Whereby how do we actually collect the data on the characteristics that we want to investigate. So all the solutions need to be tailor-made even more specifically for that particular drug that you're going to investigate on. So again, before we do or we design the study in a way, the easiest way is just to check the published papers and obviously to check the guidelines as well, because sometimes they do specify what do they need for certain drugs or certain ingredients that we use. Let's start with the easiest one again, right? So the easiest general one is about the parent compound. So i.e. the drug that you take in per se. So that's the parent compound. Whether it's active or inactive, that will be our main substance that we measure or we evaluate to be a surrogate or as a marker for the Cmax and for its distribution, because that's the first thing that comes in. So that would be the ideal marker to show that how the concentration fluctuates around after all the absorption and so on, distribution in the body, right? Because obviously metabolites, there's more difference or variance across different regions or different individuals, regions, i.e. their ethnicity and all, with the difference in the metabolizing CYP enzyme and so on. So Cmax of the parent compound is normally the most sensitive one to detect the differences. So we'll try to use the Cmax of the parent compound as much as possible. Next, we'll look at inactive prodrugs. So again, we'll try to use the parent compound again as much as possible. But sometimes, if the prodrug has very, very low concentration, or either it's just being metabolized or eliminated really, really quickly, that you could not really catch the Tmax or Cmax quick enough, right? So it is okay to actually measure the concentration of the main active metabolite, right, without measuring the parent compound, right? So in a way, in the context of this, so that's why you assume that your parent compound is an inactive one, or it actually doesn't contribute a lot to the clinical efficacy, right? So bear in mind, if we actually measure the main active metabolite, so you probably expect there will be more variations in the data actually, because again, there's issues, or there's more confounding factors that there's within the context to influence the overall bioavailability, right? Which includes, as mentioned just now, your liver metabolizing enzyme, the pharmacogenetic differences and all, which is going to be a little bit more tricky to get BE. So in this case, another circumstance whereby we need to use the metabolite data is because that the sensitivity of the analytical method for the parent compound, so that we just cannot make it at a good enough detectable level. So therefore, we have to use the metabolite data. Right, so again, if we are not using the parent compound as the main marker that you're measuring for the concentration, so again, you have to show proof that this is the only way that we can do it, right? And the method that you use, this is good enough, and so on and so forth. But bear in mind, again, as any exceptional cases, you need to back up, and there's also something that you need to prove, whereby if you have any data to show that the metabolite concentration will reflect directly or very similarly to the parent drug, and it does not saturate at therapeutic doses, then it'll be good enough. Okay. In this other set of scenario, whereby we need to tailor-make the protocol to obtain your characteristics to be investigated, is about enantiomers. As we all know, there's certain very few drugs whereby we have enantiomers, like the R or S stereoisomer, actually produce very different pharmacokinetic profiles or very different toxicology profiles as well. So most of the time for the drugs, they are quite similar in terms of the R and S. So we normally just combine them. So we use achiral bioanalytical methods, right? Whereby we just combine the data on their concentration. However, under very, very rare examples, that the individual enantiomer concentration will have to be measured separately. So the guidelines actually specify 3 particular concentrations, and whereby all 3 criteria needs to be fulfilled. So it's not that we just need to fulfill 1 or 2 criteria only, but it has to fulfill all 3 of them. So what's the 3 of them? The first one, the enantiomer exhibit different pharmacokinetics. So maybe the R, just an example, the R stereoisomer, he has reaches the Tmax and Cmax very quickly, and it goes up very quickly. Maybe the S one, just stereoisomer, just goes up very gradually and goes down and gets eliminated very slowly as well. So that is very, very different. So the second one is they exhibit pronounced difference in pharmacodynamics. So maybe they bind the target differently, for example, in the different binding strength, and they produce very different efficacy, right? Or even very different toxicity profile, because they bind to different enzymes selectively. Who knows? And the third one is about the AUC ratio of the enantiomer is modified by a difference in the rate of absorption. So it's again can be very different. So again, this will be spelled out in terms of which drugs that fits into these 3 categories. that fits into these three criteria will be spelled out in the guidelines as well, when you actually check out the individual drugs, and then you know what you need to do, right? So in most of the cases over here, it's enough to actually, sometimes if the other one is active, another one is inactive, so it's sufficient actually to just do BE study analysis on the active enantiomer only. So whereby your sampling time, design and all will just be based on the profile of the active enantiomer. So remember, at the sampling time whereby it's near the peak, and Cmax and Tmax, you need to get more sampling time around that time frame. So you just need to consider the active enantiomer if the inactive one has not much activity towards the clinical efficacy. A little bit more information on the urinary data that was mentioned earlier on. So you just have to be very carefully justified. That's thing number one, because remember, there's a lot of better options that you can use, including the measurement of metabolic parent. If it doesn't work, you measure the metabolite and so on and so forth. And you can use different types of machines, to measure what you need. Unless you really need to go to the urinary data, you have to be carefully justified, and you have to estimate the peak exposure, which will be a little bit more tricky. Again, there's other factors that comes in, whereby your urinary function, how fast does a person produce urine excretion, so the renal profile and things that will come in as well, so it'll be more complex. So again, this is our very last resort actually. In the guidelines, the EMA or the ASEAN one, you can see that they actually spend quite a lengthy section to explain the additional steps and precautions, and allowed exceptional steps that you can do when you study this category of drugs, which is under the endogenous substances. So as we already know, there's baseline level of hormonal levels for everyone, so you actually need to do baseline correction, right? That's the first thing that you need to do so that you know the exact effect of the drug that you top up, right? So sometimes there's obviously different dose of a substance that you actually develop. So, and under certain circumstances, it's actually allowed to actually use a supra-therapeutic dose, which means that in the study you can actually use a much higher dose than what is given what you design, provided of course the dose is well tolerated, and all, so that the additional supra-therapeutic dose will allow the concentration to be more measurable. That's the main purpose. As mentioned earlier on, those in the clinical setting, those who actually need endogenous substance are actually those who have a deficiency in it. So that's why the additional minute would be sufficient for them. But for normal healthy volunteers, it could be tricky to measure the differences, right? So if you're trying to use a different dose and things like that, so that is not being established, or you couldn't find any literature which support the dose or whatever that you're trying to plan or to do, it's actually allowed that you can actually do a pilot study first to see whether is it feasible to run the study, or is it sensitive to actually detect the potential differences between the formulations that you need to measure it. A little bit more details into the endogenous substance about the correction. So the exact method of correction should be specified in the guideline before it's being done. So the main general easiest way is that there's a subtractive baseline correction, basically just minus off the baseline, but you actually need to decide whether the subtraction is based on the person, right, each individual, or it's based on the mean of the individual that you're looking at. So this is something that you have to decide first, right? So this is the preferred method. And another thing which is measured, another point to take note about, is the carryover effect, which is mentioned early on in the washout period, right? So we actually need to take extra care to make sure that the effect that you give for the second dose is not a carry forward from the first one. So the easier way again is just to space it out a little bit longer. So thank you everyone for staying with me until now. So we're going to go through the last section for this particular video, which is on the strength to be investigated. So how many doses do you need to test? So again, this particular section is mainly tailored towards the immediate release forms. Later we'll go through a little bit more detail about the modified release as well. So do we need to test all of the strengths that we are going to market to our patients, right, or to our customers? So when we try to limit it to 1 or 2. That's the simplest way. So again, it depends on the proportionality of the composition, because obviously when you put it a higher dose, the concentration of the active compound in that particular capsule or the tablet will increase, right? So how the later section of this guideline, we actually elaborate a little bit more about the criteria that define the proportionality, whether the linear proportional increase, that we're going to look about it later, right? For the concentration of the different excipients as well, like your glidants, your fillers, your disintegrants, and so on. So the strength to evaluate depends on the linearity in the pharmacokinetics of the active substance. So most of the time, it will be in the linearity. Fingers crossed in a way, so that you just need to measure or just to do one BE study for that range of the same active ingredient that you want to market or to register. Right, so the details of the criteria, again, it's mentioned that it's considered to be linear if the differences is no more than 25% dose adjusted mean AUC, right? And sometimes under different criteria, you can actually consider a biowaiver as well. So again, details we'll look through at the next few slides. So ideally, the strength that you choose to do a BE study will be the strength which is most sensitive to detect a difference between the different products that you're gonna measure. So therefore, you can happily or strongly support and say that, hello, I should be qualified for a biowaiver for the other doses, right? Now let's look more into the criteria for the biowaiver that you can claim, right, for the other doses that you didn't do a BE study. Okay, so there's a few criteria. So there's 4 main criteria, but the third one, which is the C part, is really long, so we'll look more into the next slide. So the first one is actually the same manufacturing process. Second one, the qualitative composition of the different strength is the same, right? So, and the third one over here, which is the D, so you have to give in vitro dissolution data as well, so the IVIVC data, right, that you need to do for all the strength, but for BE study, you just submit for 1 dose, right? Let's look at the third one in more detail. So I didn't highlight things over here just because it's quite a lot. Right, so some parts are highlighted, but a lot of it are main points anyway. Right, so the compositions of the strengths are quantitatively proportional. Right, so when you look at the detail, what it means is that the ratio between the amount of each excipient to the active substance, the ratio should be about the same. So, but it does not cover things like, your color agents, flavors and things, which is not the main part that will affect your dissolution rate, right? So that's the science or scientific theory behind it. Right, so another part is that the C can be considered fulfilled if you fulfill criteria 1 and 2 or 1 and 3. So 1 is the most important one. So when we look at the details of 1, so the first one says that the amount of active substance is less than 5% of the overall tablet weight. Okay, so the easy way is you just need to calculate it, right? There's some maths. So the second criteria says that the amount of different core excipients, right, or capsule content are the same for the constant strength, and only the amount of active substance is changed. So your other components like disintegrants and stuff will still be the same, because the overall active substance is less than 5% of the overall weight, but you just change the amount of the filler to top up or to make sure that the weights are the same because of manufacturing easiness. Right, so number 3, which complements with the second point, because there's difference in the weight. So how do you change which is the ingredient that you can play around, right, to make sure that, it's the standard 500 mg, for example, for the manufacturing process. So it's the amount of filler that can actually account for the changes, but the others, the ratio and all should still be the same. So that's the N for the first and the O for the second and third one. So the easiest way is to actually create a table for the weight and calculate the percentage of all the ingredients. So you actually need to list down the name of each of the ingredients and what's the weight of each of it inside. So you calculate the percentage of all. Then you would know that your particular formulation, or that which is the dose A and dose B. So can you get a Biowaiver for dose B, for example? So this will provide the answer. Now let's go back a little bit to the linear pharmacokinetics, which maybe it sounds a little bit confusing earlier on. So in this particular subsection, when they talk a little bit more about it, so it actually tallies a little bit with the one just now when we talk about the Biowaiver. So you need to fulfill conditions A to D, right? So the 4 main conditions, right? So it is sufficient to just do one BE study for one strength. So ideally, so you just do it on the highest strength, right? So, but under very rare exceptions that you can do at the lower dose is preferred rather than the higher dose, is whereby if the drug substance is highly soluble, or that's due to tolerability or safety issues towards the healthy volunteers, then you can tell that would be the evidence or to back you up to say that I will only do one strength, but not on the highest, but on the middle range or the lower strength. In this slide, we'll actually elucidate another rare circumstance whereby the sensitivity of analytical method is just something you just cannot change. So therefore, you actually need to use a higher dose than what you actually market, right, to make sure that you can actually get the concentration. You can actually analyze the blood samples to get the concentration that is measurable. So this is when you actually use a higher dose. So how do you do a higher dose? You can actually use multiple tablets of the highest strength. But again, you have to back it up with safety data. Obviously, you don't want people to fall very sick after giving ultra high dose. So you must make sure that the safety data is available and there's no absorption or solubility limitations at this dose, because bear in mind, some drugs actually rely heavily on active transport for its absorption process, right? So if it's under dose circumstance, so you actually give a much higher dose, you don't really get a much higher concentration, blood concentration profile that you can measure. So there's no point of using a higher dose then. Now let's move on the other opposite spectrum, whereby from the linear to non-linear pharmacokinetics, it actually uses the same parameters or same principles as the earlier one, whereby we try to do the BE studies to be conducted at the highest strength available, so that produces the most reliable data in a way that, and it's also most of the time it should be sensitive enough to be measured by the machines that you're going to use. However, based on the name suggests it's non-linear, so the proportional increase in the AUC may not be proportional, of course, with the increasing dose. So therefore, we actually need to apply something called like a bracket approach. So it's like how you draw a bracket. So you have to do both, 2 doses in this case, whereby the highest strength and the lowest strength that you actually need to market it. So there are also some exceptions available. So if the non-linearity is not caused by limited solubility due to, as mentioned, like saturations of active transporters, and you also fulfill the 4 criteria that were mentioned just now, and you don't actually add in any excipients that may affect, the gastric motility or transport protein, you can actually just do one study at the lowest dose or the strength at the linear range. It's still okay in a way. All right, so these are some of the exceptions available as well. So a similar concept like the non-linear one, whereby we use a bracket approach. So there are sometimes we actually specify in the guidelines which drugs you actually need to fulfill, or that falls into this category. So you actually need to use the highest and the lowest strength, or the two strengths which differ most in the composition. So most of the time it's still the highest and the lowest, because the percentage changes in the the active ingredient. It may be more than 5%, for example, and so on. So this is when the bracketing approach will come in as well. But they also give some leeway in the way that, as we know, some drugs, if you still remember the class a long time ago, some drugs, or most of the time, you actually measure it in fasting state. But for some drugs, you need to do the fat state as well. So I mean after food, or together with food in a way, and how the drug absorption changes. So if the drug that falls into the bracketing approach, that is also falls into the fasting and fat state approach, it's actually sufficient to do only one strength, right? So that you don't have to do so many BE studies just for one drug, right? Because in a way it's non-economical, and in a way you actually expose your participants to multiple doses of the same drugs, because bear in mind, obviously they are healthy volunteers, so it's a bit unnecessary and unethical to do so many of it. So again, if you want to cut down to one drug or one BE study, for not really one BE study, two BE studies, because there's fasting and fat state for one dose. So again, you need to provide data from previous studies and things like that. So again, it's something back to the point that you have to dig out your journals and things like that, so you can see what people has done or has reported about the drug. Last but not least, for the immediate release preparations, as we all know, there's a lot of drugs which are marketed as fixed combinations now. For example, for antihypertensive, so you can just mix the drugs from the diuretic groups plus ACE inhibitor or ARB and things like that. So they are known to be safe. They are known to combine to a pill to improve patients' compliance and things like that. So if you want to do, you can do such formulation for a generic drug as well. But you need to, when you do your BE study, you actually need to view them as two separate entities in a way, because obviously you need to measure two particular drugs, right? So when the proportion composition should be fulfilled as well. So remember the percentage changes and so on. So you have to apply the same criteria, right? So if you consider the amount of each active substance, if it's in a fixed combination, the amount of each one, so the other active substance, in a way, it can be considered as an excipient, so therefore you just measure one concentration. So in the case of bilayer tablets, so each layer, if you use bilayer tablets, so you consider them independently, so again you actually need to measure two different drug profiles in the same. You can do it in the same BE study, of course, because the person will be swallowing this one tablet, right, that consists of two drugs, but you need to do the separate analysis for the active ingredients most of the time. So now let's move on to the very last section, just a very tiny section, just bear with me, about non-immediate release formulations, right? So i.e. most of the time it's under modified release. As we know, for modified release tablets, in a way, sometimes the drug might be more prone to hydrolysis in the stomach acid, for example, and things like that, so that you want the drug to be released later in the gut. So therefore you formulate it in a non-conventional way, so they might swell, and things like that, so that, or they can disintegrate, the outer layer at the later part of the gut, of the GI tract, in the small intestine. So these are all the immediately release, i.e. modified release ones. So there's actually specific guidelines that that determine the what you need to do or under different cases, right? But a lot of times, sometimes there'll be biowaivers and things like that, which are mentioned in the guideline as well. So if you deal with such, so make sure you just check the guideline very carefully. So there's a section again on orodispersible tablets. As we know, it's one of the hip thing about formulation, because you can quickly put it in the mouth and it dissolves and things like that. So it's much easier in a way in terms of administration. Right, so but here, when do we need to do BE for this? So when it can be demonstrated that the active substance is actually not absorbed in the oral cavity, so you just put it in, but eventually it still, the drug itself still need to be swallowed and absorbed in the GI tract, you may need to still do a BE study. But then the product may be considered for a biowaiver as well, right? So depends on which category of it that it's in, that you can actually check more information under Appendix 3. So it's actually a very long description. So you actually need to see which drug you are dealing with, and then you see which category it can fit into. So over here I actually won't explain over extensively on this section. Right, so you can see whether should you take it with water, without water, during the BE study itself, right? So if you cannot demonstrate all these things, you need a BE study. So there's different criteria again, right? So for the study design that you need to keep an eye on. So if you try to make your BE study, the way of administering the drug to best resemble the intended use of the formulation, right? If it needs to be taken with water, taken without water, so depends on how, the drug company wants it to be, advised to the patient. So we actually have to tally that in a way, right? So again, we try to do the same condition again. Everything else is just to mimic the same one as earlier on. Right, so if it's without water, so it's still recommended to wet the mouth, right, by swallowing a little bit of water, 20 ml of water directly, then to wet the mouth, and you quickly apply the orodispersible tablet on the tongue. But within an hour of it, you should not drink any more water, so to make sure you get a clear idea on how dispersible the tablet is. Right, so all the other formulations like buccal tablets, phlegm, sublingual, chewable tablets should be performed the same way, for the starting part of it, but the later part of BE analysis will still follow the same principles as the earlier ones. So in this last section for this video, I'll talk a little bit on oral solutions. So this is just to highlight some of the exceptions in a way, right? So most of the time you can get a BE study be waived, but if you actually use excipients that may affect the GI transit, like sorbitol, mannitol, right, some absorption stuff, that can affect the transport protein, affect the in vivo solubility, and things, or instability, you actually need to do a BE study, right? Unless you actually justify, the concentration or the amount that you use is okay, it won't actually affect the absorption process and so on. So this is just to highlight. So under certain circumstances whereby when you use things that you cannot actually assume that the release profile will be, as normal, there'll be changes in the unexpected or expected changes in the release profile, you actually need to prove that the product is still bioequivalent. So that's the whole scientific reason behind this. So this is just to highlight this point. Over here, when we talk more details on the modified release dosage form, so you can see the EMA has quite a guideline itself again. So the main part of the guideline actually is trying to tell you under different scenarios, which dose do you need to choose to be investigated, right? Because we all know modified release, it may result in different absorption profiles. So it may affect, again, that would result in a different curve that you may get in the end. So this particular chart is from the guideline as well. So sometimes the single dose that you choose, so you normally choose the highest or the most sensitive ones. Then we go down that, if we really have to do, if we can't get a good data in a way which is supported, by other studies and all, you may need to use multiple dose, right, and things like that. So again, this is actually quite a thicker guideline by itself. So if you actually need to do more, you actually need to do or design a study on modified release, I suggest you to look more into the guideline to see which the drug fits into which category of it, that which points that you need to take note about. Conclusion. Thanks for staying with me. So the main thing that the take-home message for this whole particular video is that when you actually design a study, the easiest thing is, the first thing is to check the authorities, all these guidelines and things like that, because sometimes certain drugs will be specified in terms of what you need to do specially about them, when you do your analysis or during your design. Right, and another part is some stuff like modified release, when you have it's also governed by other specific guidelines as well that you need to double check as well. And thirdly, always check the journals as well. So sometimes, or most of the time for most drugs, you can at least find 2 published journals that tells you what you can do or how you can design the study for the drug, because you need to know also, for example, like what bioanalytical tool that they use, right, to see whether is it available in your facility, for example, about the costing and things like, can you replicate the study or use that as a, you may not replicate exactly, but you can use it as a good reference as well, like when is your Tmax and so on. How do you analyze it? What column do you need to use, and things like that? And what's the rough, concentration or maximum concentration that we get with your, or the peak that you get with your, with the concentration standards that you use, and so on and so forth. There's a lot that you can learn from other studies in a way. So first thing, when you know you need to do a B study on drug A, check. Right, so that will save you a lot of work as well. Last but not least, thanks a lot for watching the video, and thanks a lot for staying until the end. So if you like the video, if you think it's informative, it's helpful for you, do subscribe the channel, turn on the bell so you don't miss the other videos, and also give a like to the video. Hope to see you again soon. Ciao.